textbook readings are definitely testable on the exam. However, the focus of the exam will be on lecture-related material. So I'm not going to stand up here and lecture for like 3 hours a week if I'm not going to test you on this stuff, if this is not the important stuff, right? I'll just go home and watch TV, you can go watch TV, right? Please relax. But we're here because this is the important stuff. So make sure you know the lecture material, take a look at the textbook readings, they are testable, but make sure you focus in on the stuff that's really closely related to the lecture material. So just to give everyone an idea for what everyone else does, let's do this survey. So I think it really helps students to see what everyone else does. And different students will be different, right? So let's just read through these and then we'll do a quick survey. So which of the following best describes you? And again, different students are different, so let's just see what everyone's at. And this will give people who didn't take Bio 130 an idea for what's going on. So A, I did not need to read the textbook to do well on the Bio 130 exams. B, I needed to add to my notes from the textbook readings to do well on the Bio 130 exams. C, I need to memorize every detail from the textbook readings to do well on the Bio 130 exams. Okay, so A, who did not need the textbook, right? So raise your hand, everyone look around, right? So a lot of people didn't use the textbook. B, who needed to add to your notes from the textbook, right? Raise it high, or everyone look around, right? And C, who went crazy, who really relied on the textbook. Lots of people do, right? So you can kind of see the distribution. And students will always ask me, well, what did you do, right? So again, different students are different, but for me, I was somewhere in the middle. So how I did it as a student is I would always read the textbook before coming to class. I always found that if you go into the class kind of blind and just learned it for the first time, you missed a lot of the nuances, you missed a lot of the emphasis that the professor gave, and maybe you missed some of the hints. So I always like to read the textbook beforehand. I'd go to class, take lots of notes, then I'd go home, read the text, and then add to my notes. So that's what I did, and then I'd study for my notes, do practice exams, right? That's the kind of thing that I would do. So hopefully that helps. All right, so let's begin the lecture material. So what I'm going to tell you here is on the next slide, and you're going to write this all down on the next slide, so you really don't need to jot down too much right now. So the tree of life is shown here. It has 3 primary branches. You have the eubacteria, archaea, and eukaryotes. The stuff we normally call bacteria is kind of in the eubacteria family here. Both eubacteria and archaea are, of course, prokaryotes. They lack a nucleus. Eukaryotes include plants, humans, they have a nucleus. Archaea tend to live at very extreme environments, and because they're prokaryotes, a lot of people tend to group these two together. Say, oh, they're prokaryotes, they are very similar. But if you kind of look at when they diverge from a common ancestor, the eubacteria, the archaea, and the eukaryotes all diverged at around the same time. So yes, archaea, they're missing a nucleus. Yes, eubacteria are missing a nucleus. But in many, many respects, archaea are actually very similar to eukaryotes, especially when it comes to stuff like gene expression. So that's something that we'll talk about coming up in this particular course. So don't worry, I know this already, but you'll have the notes that you need to know on the next slide. So this is the tree of life. That's in your textbook, that's in your e-text, as well as in your lecture notes. I just want to show you kind of a more accurate tree of life. It's actually, of course, much, much more complicated. It does look something like this. And I just hope, like, nothing to memorize here, right? So I just hope that as you go on, that one day you guys will be able to play around with some of these databases and kind of zoom up on the real life tree of life. So a lot of scientists have painstakingly mapped all these different species, and you can actually play with these databases and zoom up on the current existing species, and then kind of go back and track the various other types of species that people have found. Okay, so what do you need to know? There are 2 main cell types, prokaryotes and eukaryotes. Prokaryotes include eubacteria and archaea. They are single-celled. They lack a nucleus. They lack organelles. Correct. So prokaryotic cells, they're single-celled. They lack a nucleus and organelles. Eukaryotic cells include plants, fungi, animals, and humans. Be careful here. A lot of people mess this up on exam questions. Eukaryotic cells, single-celled or multicellular. So yes, prokaryotic cells are single-celled. Eukaryotic cells, single-celled or multicellular. About half the Bio 130 students actually screw that up, so be careful. Don't screw it up in Bio 230. Eukaryotic cells have a nuclei and they have organelles. So first off, we have your standard prokaryotic cell. Let's go through some of the cell parts. You have a plasma membrane. So it's a selectively permeable filter. Selectively permeable filter. You don't have to write this down, right, if you know it cold. So selectively permeable filter. DNA, right, genetic material, and it's present in what we call a nucleoid. A nucleoid. Hope in Bio 230, no one's furiously writing DNA genetic material, right? Just insurance, just for insurance. Okay. You have DNA, it's present in a nucleoid, it's genetic material. So it's not a nucleus, but it's in a compact structure called a nucleoid. You have a cell wall. This is optional in prokaryotes. Optional. Tough, protective outer coat. Tough, protective outer coat. Tough, protective outer coat. Ribosomes, protein synthesis. Right, everyone knows that? Don't let your neighbor see you writing that protein. Okay, flagellum is optional for locomotion. Optional locomotion. Okay, so here is your standard prokaryotic cell. Plasma membrane, selectively permeable filter. DNA, the genetic material, it's in what we call a nucleoid, compact structure. There's no nucleus, but it is compact. Cell wall, it's optional, tough, protective outer coat. Ribosomes for protein synthesis. Flagellum, optional for locomotion. Now, if you take a look at the size here, you don't have to memorize the size, but just take a look at the size here. That's about 1 micrometer. That's for one cell here. If you flip over to the eukaryotic cell, take a look at this scale here. That's 5 micrometers. So eukaryotic cell is much larger, right? Much larger. And if it's much larger, you need a cytoskeleton, right? Microtubules, part of the cytoskeleton. Cytoskeleton. You have organelles such as the mitochondria, right? Mitochondria for ATP generation. Organelles like the mitochondria, ATP generation. So mighty mitochondria, right? It's a power plant. Right? You have the nucleus, right, where the DNA is. As well, you also have stuff like the plasma membrane that is also in prokaryotes. Okay. So again, if you take a look at the size, you can see eukaryotic cells are much larger. You have a cytoskeleton. Microtubules are part of that cytoskeleton. You have mitochondria, various organelles in eukaryotes. You also have a nucleus, and you have the plasma membrane, which is in both cells. Now that's your eukaryotic cell, and just to show you, I'm supposed to show you this quick video, show you some of the processes that eukaryotic cells have, and we showed you some of this last year, and eukaryotic cells will often go through phagocytosis.
Neutrophils are white blood cells that hunt and kill bacteria. In this spread, a neutrophil is seen in the midst of red blood cells. A Staphylococcus aureus has been added. The bacterium releases a chemoattractant that is sensed by the neutrophil. The neutrophil becomes polarized and starts chasing the bacterium, which, powered by its flagella, swims in a random path, seemingly avoiding its predator. Eventually, the neutrophil catches up with the bacterium and engulfs it by phagocytosis.
That was the video. When someone wrote in and said, oh, that was really disrespectful burping in class. Now another thing that I want to show you was this kind of video that everyone showed in Bio 130. So it's just kind of a review of everything that happened in Bio 130. So if you took Bio 130, you guys know and have seen this video a number of times. So it's that video where scientists at Harvard got together with artists and they animated as accurately as humanly possible what goes on inside the cell. It's actually very accurate. So this is a nice review of everything we talked about in Bio 130. I know a lot of you have seen this a number of times. So here we're looking at the blood vessel, remember? And you have a lot of red blood cells flowing through the blood vessel. So on the surface of the cell, you have a variety of different adhesion proteins that help the cells stick to one another. And this is the surface of the cell containing the plasma membrane. And you have a variety of different proteins on the surface of the cell. They all interact between various cells. Now you're kind of looking at the inside of the cell where you have a variety of different actin filaments making up part of that cytoskeleton. So this will be the cellular lamina, kind of the meshwork that's just underneath the plasma membrane. Here's a forest of actin. It's the cytoskeleton that gives those cells that structure that you see. And actin filaments, of course, can be constructed, right? They're quite dynamic. And you'll have also a variety of different proteins that will help modify the actin, so say the cell can crawl or cell can change shape. Here's microtubules. You're going to transport a lot of things along these microtubules. Here's microtubule destabilization. You can take these things apart as well. You'll remember the walkie guy, right? So walking along the microtubules, transporting a variety of different nutrients, say from one side of the cell to the other side. Here's transporting some vesicles. You have a variety of different organelles, and coming into focus in the background, you have the centrosome, which helps organize those microtubules. Now this is the nucleus, and streaming out of the nucleus you see RNA. RNA will circularize, and you have these unique ribosomes binding to the RNA, making this yellow protein. Proteins, of course, will localize, they'll go to different locations, such as the mitochondria shown in the background here. Here you have a ribosome making proteins to be secreted, so it's making it into the endoplasmic reticulum. Various organelles will form vesicles, and you can see vesicles can be transported again by this walkie guy. The dynesins or the kineins. Here's the Golgi apparatus. Modifies proteins, will help move them around. And all of that is going on inside this one cell. You can see it will secrete a number of different proteins to the environment. As well, it will have different membrane proteins to interact with the environment. And here it's interacting with the extracellular matrix. And all that is going inside that one white blood cell, signaling that one white blood cell to leave the blood vessel and say, go attack some bacteria. Alright, so that was Bio 130 in a nutshell. To start off this course, we just want to review a genome. Now, all known life forms, of course, possess a genome. It encodes the information to construct and maintain an organism. Most genomes are made up of DNA. Most genomes are made of DNA. Some viruses have RNA genomes, and textbooks and people often get into this debate and they'll say, well, viruses are not alive, right? Yeah, they're not cells, they're not alive per se, but they do have genetic information, and sometimes they store it as RNA. Not always, there's DNA viruses, but there's some RNA viruses as well. The release of biological information stored in the genome requires genome expression. So DNA is great, but you gotta get something out of the DNA. You gotta get genome expression. The first product of genome expression is the transcriptome. The transcriptome is the repertoire of RNA molecules present in a cell at a particular time. So you take any cell, you want to look at the transcriptome, you're looking at all the RNA molecules it has. So that's a transcription. Now, a DNA microarray gives you a snapshot of the transcriptome. DNA microarray gives you a snapshot of the transcriptome. Take a picture of it. DNA microarray gives you a snapshot of the transcriptome, which is the repertoire of RNA molecules in the cell at a particular time. Now, right now, you don't need to go crazy and look through all the different mechanisms and techniques used to do a DNA microarray. You just have to know how to read a DNA microarray. So if you look at this, how do you read a DNA microarray? Well, it's basically a table. It's basically a table. So the rows here are essentially the genes, right? So genes, so different rows, gene X, gene Y, gene Z, etc., 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 right? Very small, but all the different rows are the different genes. And all the different columns are the different kinds of cells. So here's one liver cell, here's another liver cell, here's another liver cell, here's a breast cell, here's another breast cell, etc., right? So all the rows are different genes. All the different columns are the different cells, or the different samples. Now, red means lots, and green means very little. So red is lots, green is very little. Red is lots, green is very little. Okay, so let's say we look at this row, and I know I've circled a lot here. It's pretty magnified. So say we just look at this particular row. Say this is row Y, right? So you're looking at gene Y, and so you can see over here in the liver, it's more red. So liver cells have lots of that RNA, lots of RNA for gene Y. And maybe, let's say, brain cells, it's green, so they have less. Right? So red is lots, green is less. Okay, to summarize, from the genome, first thing you get is the transcriptome. That's all the RNA molecules present in a cell at a particular time. A DNA microarray gives you a snapshot of the transcriptome. What you need to know how to do is to read a DNA microarray. It comes up again later on in the course. So to read a DNA microarray, you just read it like a table. The different rows are the different genes. Different columns are the different samples, like different breast cells or different liver cells. And all you do is read it like a table. Red means lots, green means little. So if you look at this row, for example, let's say this is gene Y. Well, liver, lots, it's more red. And then say brain, well, less, right, more green. And that's simply how you read it. Lots of the RNA versus less. Okay? Just read it like a table. Now the transcriptome is maintained by the process, of course, of transcription. The second product of genome expression is the proteome. So just like the transcriptome is kind of all the RNAs, well, proteome is all the proteins, right? So the collection of proteins in a cell defines the biochemical functions of a cell. Now, just like a microarray gave you a snapshot of the transcriptome, a 2D gel electrophoresis gives you a snapshot of the proteome. 2D gel electrophoresis gives you a snapshot of the proteome. Now, to read it, you know, microarray, we said it's like a table. Proteome is kind of like a graph. Okay, so you actually have two 2D gel electrophoresis here. One for the human brain, one for the human liver. So you kind of divide there's two of them. The y-axis is essentially molecular weight, right? So high weight versus low weight. And the x-axis is isoelectric point. So you can read more about isoelectric point if you're so interested. It's kind of like pH. Not the same, right? Chemists will be like, oh, no, it isn't. But you can kind of think of it like pH. Okay, so that's essentially how you read a proteome. Y-axis is molecular weight. X-axis is, you know, kind of like the pH. Now, here the different blobs represent to you different proteins, and they're colored in for you here. On a real 2D gel electrophoresis, they're not colored for you. Okay, so red and blue is colored for you. Red shows you the same protein. So let's say here's a human liver. Again, y-axis is molecular weight, x-axis isoelectric point. So if you look at this blob right here, fairly high molecular weight, kind of acidic, well, it's essentially this protein right here. And so red just shows you the same protein. Now, if you take a look at the same protein, yeah, the human brain can definitely have the same proteins as the human liver, but look, the protein here is at different amounts, right? The blob is bigger here, the blob is smaller. So they can have the same proteins, but they can be there in different amounts. In addition, you have in blue the proteins that are unique. So if you look here, this blue protein is in the brain, but the same spot, it's empty in the human liver. So sure, they'll have different proteins as well. All right, to summarize, second product of genome expression is the proteome, all the proteins. To get a snapshot of the proteome, it's a 2D gel electrophoresis. You read it like a graph. Molecular weight on one side, you can just think of this as pH on this side. You can see that it's color coordinated for you, but in real life it's not going to be color coordinated. Red shows you the same protein, so yeah, the brain and the liver, they may have some same proteins, but they can be there in different amounts. As well, in blue, they show you unique proteins, like proteins that are only in the brain, not in the liver, and vice versa. So some same, some unique. The proteome is, of course, maintained by translation. When we talk about going from DNA to RNA to protein, that is the central dogma of molecular biology. Different cell types of a multicellular organism contain the same genome, but they can be different because you have differences in genome expression. So here you see a picture of a neuron. So think of one of your brain cells. Here is a lymphocyte. Your brain cells, your lymphocytes, they are vastly different. They look very different. They have very different functions. Their genome is the same. They're different because of differences in genome expression. Right? Your various cells, all the cells of your body have the same DNA, but they're different because of what comes out of the DNA. So the human genome is about 25,000 genes. At any one time, only 30 to 60% of the genes are expressed. Expression of almost all genes vary from one cell type to another. This is the same 2D gel electrophoresis. We don't need to go through it again. Red are proteins that are common. So again, remember, brain and liver, they'll have some proteins that are the same, but they'll be there in different amounts. As well, you have some unique proteins, some proteins that are only in the brain or only in the liver. Genome expression is regulated at many steps from DNA to protein. Again, different cells, same DNA, but you change what comes out of the DNA. So here is an overview of the central dogma. DNA, you know, can replicate. You can change its organization. The red lightning bolts show you all the different ways you can change what comes out of your DNA. Right? You can change how compact DNA is. You can change what RNAs get transcribed. You can change how the RNAs are modified, how they're spliced. You can change how much it gets translated. You can post-translationally control proteins. You can modify proteins. You can sort proteins to different locations. You can change how they interact. All of that affects the interactome. Some of you will remember that's the variety of interactions in a cell, all the different interactions of, say, proteins. And the interactome, all the different interactions of the proteins, will affect the metabolome, all the small molecule metabolites in the cell. All those small molecule metabolites in the cell. So don't forget, your cell is not just DNA, RNA, and protein. There's other stuff, right? There's nutrients, there's glucose, glucose-6-phosphate, all the stuff with the electron transport chain. There's lipids, phospholipids, right? All that stuff. So how you affect all the small molecule metabolites, well, that's metabolism, all the other stuff. Okay, so that's essentially the regulation of genome expression. Now, I went very quickly over these red lightning bolts, all the different ways you can change what comes out of your DNA. We're going to go through these red lightning bolts throughout this course, so you don't need to go crazy taking notes right now. You'll see us go through it throughout this section of the course. So, just a couple of quicker questions as a nice review. The bacterial toxin cyclohexamide inhibits eukaryotic translation. Which of the following would you expect to be most effective in human cells treated with cyclohexamide? A, genome, B, transcriptome, C, proteome. Listen if you can. Good work, yeah, C. So C is the proteome. So if you stop translation, right, you're stopping making protein, the thing that's most effective is going to be the proteome. All right. This is a previous exam question. This is actually a question that I made. So if you don't like it, you can blame it. The bacterial toxin cyclohexamide inhibits eukaryotic translation. If a scientist wants to compare differences in cyclohexamide-treated versus cyclohexamide untreated animal cells, which of the following techniques should you use? A, DNA sequencing, B, DNA microarrays, C, 2D gel electrophoresis, D, RNA sequencing. So buzz in if you can. All right, nice. So everybody get this right if you see it on the exam, right? So I want to see this kind of thing on the exam. So it is 2D gel electrophoresis. Okay, so with that being said, let's take a 10-minute break. So I have 7:05. Let's resume at 7:15. Questions are welcome if you have any.